So at the system level, and I really wanted to talk to you about this tonight, I want to talk to you about the work I've been doing the last 2 years on pain control. I don't have time to go through all the experiments. One I'll present sort of a summary form. But based on our work, we have proposed that there are 2 different methods that we could use, and probably they're most effective if we use them together, to modulate pain, to change this chronic pain pattern, and to block pain transmission. And all of it, my lab showed, is based on the dose of light you give. So if you look at this panel, we found, and also based on the literature, that if we're under a irradiance of 100 milliwatts per centimeter squared, that we sort of turn on everything that's generally spoken of in photobiomodulation. First of all, we did in our animal models get decreased pain response. We totally changed the chronic inflammatory pattern, and it was amazing because we caused inflammation by damaging the sciatic nerve. But when you have pain turn on that changes to chronic pain, you get inflammation up your entire ascending sensory system that's maintained. And we found with the treatments we did in this range that we changed that. We started getting expression of anti-inflammatory markers over pro-inflammatory markers. And then we also did some experiments that if we upped the output power and we used 250 milliwatts and above, that we could get a rapid block of pain transmission. And this is utilizing the inhibitory aspects of photobiomodulation. It's just not stimulatory. The dose was high enough that we altered the microtubules inside the neurons, which stopped mitochondrial movement, blocked action potential, and stopped the pain. So we really are proposing, and I must say I'm very pleased because I just received a grant from the Defense Health Administration, 2 years of funding to work on these 2 methodologies. But we're proposing in cases of severe pain that we can block the pain and then go to the lower levels of treatment to take care of the chronic inflammation and break this pain cycle. So a couple of years back, a young functional neurosurgeon from Brazil did a study, and her name is Dr. Holanda. And what she did was she got patients that she put into 3 groups: an ultrasound group, the standard lidocaine injection for back pain, which these functional neurosurgeons will put in at the lumbar 2 dorsal root ganglion level. And then these Brazilian companies tend to make very low output devices, and you can see here that the average radiated power was only 100 milliwatts, but they put it through a tiny fiber, and out the tip of that fiber, it came out as 35 watts. And she put that fiber down, the same needle she used, same size needle she used to inject lidocaine, and put it not right up against dorsal root ganglion, but envisioned this under the fluoroscope within half a centimeter of it. And this top panel, I'm sorry if you think it's a little gross, but this is down in my university's cadaver lab. I teach medical students head and neck anatomy and neuroanatomy. And so I have access to the material, but here's the dorsal root ganglion. And we took our device and we measured what was actually hitting that ganglion, and it was about 4 watts. It was probably still a lot higher than she needed. But the interesting thing is the pain. She could ask her patients, what are you feeling? The pain stopped within 5 minutes, and the pain was suppressed for 21 days in these patients. She's now trying to get funding to do a longer study. The interesting thing that the pain blockage was comparable in lidocaine and comparable with the light through this fiber. but they were both much better than ultrasound. I know a lot of people are running to get ultrasound for their back pain now. So she came to my lab then that fall, and we wanted to look at the mechanistic basis of what was happening. And so here is a pretty cell, dorsal ganglion neuron in culture. And an Australian investigator, Dr. Roberta Chow, was working on also dorsal ganglion, and she published using a different wavelength that in culture, if she used 300 milliwatts per a centimeter square area, which is high, usually you're like less than a milliwatt, percentage of a milliwatt when you deal with cells, that she could get different changes in the mitochondria in her cells. And we did that, and we looked at mitochondrial metabolic activity, and then we looked at the neurons themselves and what happened to them. And so in this panel, you could see here, using this MTS assay, which looks at metabolic mitochondrial activity, that we downregulated it at all these different times. And I don't know if you could see it with the lighting, but at all these panels, these neurites that were growing out from the ganglion neurons form these bowls, like balls on them, like rosary beads, and they're called varicosities. And it's known that these varicosities are due to breaking of the microtubules. And so the functional change then, and the other interesting thing that we saw was that only the smaller neurons, because you get a dorsal root ganglion, it's different sized neurons in it because these neurons do different jobs in your body. And the smaller ones, 30 microns or less, are the ones that transmit pain and temperature. They're the ones that were affected. The big fibers, which are proprioceptive, allow you to know where your body is in space and to walk correctly, they weren't affected. And that's incredibly important because if you want to do a treatment and block pain, you want that person to keep their motor function. And in fact, that's one of the things we're doing with the military funding, is to see in a mixed nerve, not just sensory nerve, if we can block pain and maintain motor function, which we think is very feasible because motor neurons are the largest in our bodies. So I wanted, no one had done this before, and I wanted to look, and there's a point to this slide besides my measuring light transmission, although I do measure light transmission all the time. But my lab was the first to measure light transmission in anesthetized rodents. Before this, people used dead rodents, or they used Monte Carlo mathematical modeling, or they used created phantoms like fake tissue. But we used anesthetized rodents. And Dr. Sean Wang built me a special sensor where I could actually measure irradiance per area inside the animal. And I wanted to see, I had an 810 nanometer laser that I wanted to use near infrared. And I wanted to see what would I have to put on the surface to get near 300 milliwatts inside the animal. Now we're talking here, folks, about an animal with no pigment. These are albino. There's no melanin to absorb light. And look at that. It's not a big distance between the nerve that I was measuring and what light got to it. We're talking maybe a centimeter to 2 of tissue. I needed to put 10 watts of light to get 270 milliwatts to that nerve. Now, I'm telling you that because in the olden days, when this field started, and it was red light, and people were doing wounds, that was surface, it didn't matter. But then people started saying, well, let's do deep tissue. Let's do the stellate ganglion and see what happens. And you know what happened? Nothing. Because on the surface, they put the dose they were using in the tissue culture or for wound closure. And they didn't think that 30% to 40% of that light will be lost just passing through the dermis. So if you want to get light inside, you have to put a lot more light on the outside if you're doing transcutaneous application. And that's a really important point. I want to make sure we have time for questions, so I'll speed up here. So what we did was I did a spared nerve injury model, which is a very classic way to create a pain model in a rodent. You take the sciatic nerve and you cut it, and then you ligate it, and you leave one intact. And you get this big inflammatory response within the dorsal root ganglion, which makes the axons that are in the sural nerve sensitive. So now they start acting as if they're experiencing neuropathic pain. And then what we did was we took these rats, you had to wait 7 days, and we tested them. We did all kind of behavioral testing to make sure that they were acting now with heightened sensitivity to stimuli. And at that point, we did this test, and we took that 10 watts we knew we could deliver 270 milliwatts inside, and for 2 minutes we ran it from dorsal root ganglion along the nerve out to the cutaneous region. Now, we couldn't ask our rats, hey, how does that feel? We had to wait for them to wake up. So we didn't get a 5-minute response. We got an hour later response. When they woke up, we did the testing, and then we also did some long-term experiments. And the beauty of it was that for the pain pinprick test, the heat test, the cold test, that we could within an hour change this, and it was maintained for various amounts of time depending on what that sensation was. So, there's room for more research here. But the really interesting thing, just like in the tissue culture dish, we didn't affect the big neurons. So again, I thought that was just, every now and then an experiment works just like you think it does the first time, and it is the best feeling in the world. So every time I look at that, I'm just so happy. So basically, our preclinical work in animals and tissue culture shows that we could do dosing to affect pain and inflammation related to pain by doing different dosing. And I think the most effective therapy will be to combine those. And that's basically what these slides reiterate for you. Now, what if you have back pain, or maybe you have migraines or a pulled tendon, and you want to go get treated? So in the next couple of minutes, I'm going to tell you some of the things you need to think about and make sure that if you buy your own device, you're thinking about these things. Or if you go to your clinician, you say to him, what's the wavelength of that laser? Maybe they'll say, I don't know. And then you walk out the door. Or maybe they'll say, oh, well, this is a dual wavelength. And you'll say, okay, I'll stay. So you've got to think about what you want treated and where it is in your body. If it's a wound, you can use most wavelengths if the dosing's right. And in fact, red light has been shown now for 50 years to do an incredible job on healing and ulcers and burns. So it's very easy to treat, and it's very effective. But what if you have that really bad back pain? You've got to think about the wavelength and how deep it goes. It doesn't usually go this deep. I tried, but... So it's wavelength that we have to consider. And then the other thing is you got to get light where you want it, and you got to get the right dose. If not enough photons get there, it'll be suboptimal, and you're not going to be fixed. So the goal in deeper tissue PBM is to look at all these wavelengths and look what's waiting for that wavelength of light. Water, melanin in the skin, blood, oxygenated and deoxygenated. And so all these things have their favorite wavelengths to absorb. Oops, sorry. I keep pressing it instead of the... All of these things have their favorite wavelengths, and you can see their extents. Here's melanin, And you could see that just beyond melanin, there's a 1064 nanometer. I haven't worked with that much, and I'm dying to work with that wavelength because it's just beyond melanin. Water absorption is still pretty minimal, and I think it would be a very effective clinical wavelength. But if you're treating or you are a person of color, you want to be aware that, one, certain wavelengths could burn your skin because of the melanin absorption, but also the treatment would have to be different because of the melanin absorption. Because a famous guy in light tissue interaction, Dr. Stephen Jocks, has shown that if you use red light, it's absorbed 4 times more than an infrared by melanin. Okay? So these are all other considerations. So this is my famous rat. It's all over the web. I don't know how it got out there, but it's everywhere. And I go to talks and people show it and use it in a totally wrong context, but that's okay. So we did this experiment with my colleagues from the FDA, Dr. Ronald Wayne and Ilko Ilov. And they're physicists, and they were optical fiber experts. And you could see here, this is the first anesthetized rat we ever studied. And we put white light on the rat's back, and then we had an optical fiber that we could insert below the skin, into superficial fascia, into muscle, into bone, into the vertebral column, and into the CNS. And the most important thing here is we found that in the lowest layer, all the way down to the vertebrae, that there is a window between 790 to 820 with a peak at 810 nanometers that has the deepest penetration into the body. And again, our work on spinal cord injury using this wavelength has resulted in issuance of a patent for this use. The other beautiful thing we found also using live animals, and we looked at blood transmission, 810 is minimally absorbed by blood because it's right where you have oxygenated and deoxygenated blood profiles, absorption profiles cross. And also, it is minimally absorbed by water. So it's a great wavelength if you want to get inside. Now, if you use 808, that's not going to make any difference. Some people use 820, 830. A lot of that has to do with getting around patent on what they're trying to do. I want to mention to you about transcranial laser therapy. I think I'm going over my time. Shall I just finish with this? So, okay, I'm just going to tell you about this transcranial laser therapy so you know that one of the real hot areas right now is the use of light through the scalp, through the skin, I'm so klutzy, and then into the brain. And this is really important. And we tried to get some funding from like the Michael J. Fox Foundation, and they came back and said, look, no one's proven you get light into the brain. Go prove that and come back and ask us for money. And it's really a very exciting field because people in preclinical work and in some small patient studies are showing great effects on Alzheimer's, some effects on Parkinson's. Parkinson's is trickier, but great effects on post-traumatic disorder, and on other types of brain trauma. So it's got great promise. I'm on the safety and efficacy board for a study that's going on in New York and Boston right now, looking at this light for major depression that's non-responsive to drugs. And I'm so anxious to see when that data is unveiled, and that's got a good patient population. So in this study, we started to go ahead and look at the light. In this case, we just looked at brain. This is fresh human brains that we got through the state of Maryland cadaver program. And you could see if we put, this was 810 nanometer light, that the light is transmitted into the brain. And you know how the brain has these bumps and grooves, gyri and sulci? Well, look what happens. We found out that the sulci essentially act as light waveguides, and the light can get, I told you, deeper into the brain. So we went on and we did a much more elegant experiment. This was led by Dr. Clark Tedford, who is currently got fabulous data on the use of light for dry macular degeneration. Really wonderful data. And he came up with this rig where we could cut off the top of a cadaver head, fresh cadaver, cut the head in half, mount it here. We had a laser on the outside, and then these are all sensors. And basically, the bottom line is in this panel that we found that light could get 40 millimeters or 4 centimeters into the brain. Now, whether this was the optical, is a truly photonic optimal dose isn't known. Now, I want to end, but I just want to tell you the other thing using this slide is that wavelength exclusively determines depth of penetration. It's wavelength. Don't let anybody tell you anything else. It's not pulsing. It won't change it. The output power won't make it go deeper. All of that is fantasy. What you do get when you increase the output power are more photons along that given depth. And that allows you to regulate the dose you want to get to these deep tissues. There's a bunch of more studies. Yeah, I could go on forever. But I'm going to go to the conclusion, and hopefully you have any burning questions. I think that this is really, this photonic medicine is the future. And I think it, because of its mitochondrial mechanism and mitochondrial involvement in so many diseases, I think that it could address a lot of diseases we don't have a current cure for. I think that there is extraordinarily strong preclinical data. There's some indication in small clinical data, but we need a big trial to really be able to say there's strong data there. It's non-pharmaceutical without those side effects, and it's non-addicting, and it offers a wonderful opportunity for pain control. And also another hot issue, I've just been invited to actually attend an LED conference in France in November where they are focusing on the use of LEDs in the aging population, which is now huge. Both body function and keeping your muscles strong and responsive and continuing to build muscle instead of lose it, also with light, and preventing or slowing cognitive decline with aging. So those are all wonderful things.
Dr. Anders is internationally recognized as an expert in photobiomodulation, which we refer to as PBM. And in her research, she has served in noteworthy, prestigious positions. PBM is the biological response to using non-thermal, low-level light therapy to heal a wide range of injuries and diseases. Her specialty is peripheral and central nervous system injury and repair mechanisms, as well as light tissue interactions. Dr. Anders received her PhD in anatomy from the University of Maryland Medical School and then joined the National Institutes of Health in the Laboratory of Neuropathology and Neuroanatomical Sciences, known as NINDS. She is affiliated with the Uniformed Services University Health Sciences as a professor of anatomy, physiology, and genetics, and professor of neuroscience. Dr. Anders serves on the executive councils and scientific advisory boards of numerous international laser conferences. She is the past president of the North American Association of Laser Therapy, a founding member of the International Academy of Laser Medicine and Surgery, and past president of the American Society of Lasers in Medicine and Surgery. She has recently been appointed as a board member of the International Society of Lasers in Medicine and Surgery and currently serves as director of the Optical Society of America Photobiomodulation Technical Group. She is a senior editor of Photomedicine and Laser Surgery, associate editor of Lasers in Surgery and Medicine, associate editor of Lasers in Medical Science, and on the editorial board of Physiotherapy Practice and Research. She has published over 70 peer-reviewed articles, and we are so fortunate to have this pioneer in this area of research come to Shepherd University. She is also an adjunct faculty member at Shepherd. I just want to point this out. We're very proud of that.